Welcome back to the Classics in Immunology Journal Club. This week we are in 1993, moving right along here. Started in 1798 with Edward Jenner a while back. The paper we're going to review today, I guess before we get started, I want to say, please go online to the YouTube channel where you are to watch this video and like it. Click on the bell to get notified for more. Subscribe to the channel and check out my website. Scroll down and check out my website on the writing tab on my website. You'll find all the classic papers, including this one. Now, this is another one of my favorites because it's one of my papers. Again, now you don't get the idea that I'm only reviewing my own papers. I'm not doing that. But it just so happens we're on a roll here in terms of trying to figure out how interleukin-2 mediates its effects on the target cell, the T cell. And this paper was authored by Carol Beadling, Kirk Johnson, and myself. And the title is The Isolation of Interleukin-2 Induced Immediate Early Genes. It came out in the Proceedings of the National Academy of Sciences in April of 1993. Now, the background on this paper is that Carol was a grad student who'd come to us from Reed College in Oregon. And the interesting thing there's a lot of interesting things about Reed as comparison to Dartmouth. One of which is that she started working on frog IL-2 at Reed. Who knew that amphibians had IL-2 too? But that's because it's very important and evolutionarily conserved. But the other thing about Reed College is that Reed is politically a very progressive liberal institution. And if you know anything about the Ivy League here on the East Coast and Dartmouth in particular, Dartmouth is a rabidly arch conservative institution. And when Carol came to enrolled in the graduate program here for her PhD, because she wanted to work with me and she wanted to work on IL-2. And I thought that was a good idea. After she, migrated all the way from the West Coast and got herself situated at Dartmouth. And later on, she told me that she thought she'd landed on Mars because it was such a different kind of a place. But to hand it to her, she just buckled down and put her nose to the grindstone and started working. Kirk Johnson was a postdoc who come to us from Richmond, Virginia. And he helped Carol a great deal in terms of working up initially the molecular genetic techniques that we needed in order to do the experiments here. And myself, where I was focused on throughout my career is that I started out as a young clinician and a hematologist and somebody who was really interested in leukemia. And so early on, in the very beginning, I decided that I wanted to work on cancer. And I think I told you before, I would tell everybody at Dartmouth that I came there in order to find the answer to cancer in New Hampshire. This paper is really a culmination of 20 years of working on trying to work on the lymphocytes, the T cells, as a model system in order to be able to get into the nitty-gritty, the internal workings of the T cell, to try to find out how the cell is signaled to begin to replicate DNA and then undergo mitosis. Because I figured if we could figure that out in the normal cell, then we could find out what was wrong with the cancer cell, particularly a leukemic cell. So that's what this was all about. In the intro of this paper, we focused down on two aspects of the T cell cycle. And we borrowed the concepts and the findings from the cell biologists who were also interested in exactly the same question, but they weren't working with lymphocytes and T cells. They were working with mouse embryo fibroblasts, the so-called 3T3 cells, that were sold as, they were ostensibly supposed to be normal fibroblasts. But as I got into the literature and read all about these cells, I found that they weren't normal at all. They were actually grossly abnormal. They had extra chromosomes, they had translocations, they had all kinds of things wrong with them. They just didn't look under the microscope like a cancer cell. They looked like a normal cell, and they behaved more like a normal cell than a cancer cell in that they didn't really grow continuously and constantly. They would reach a point in their growth where if you seeded them very lightly into a flask and then looked at them under the microscope, you'd see a cell here and a cell there and so forth and so on. And so over the course of a week or so, those cells would proliferate and they would come into close contact with one another. And at about that time, they would stop growing. That was called contact inhibition. And it was thought to be one of the characteristics of a normal cell versus a cancer cell, because cancer cells, malignant cells, would pile up and grow and keep on growing and so forth. So that was the contact inhibition that the 3T3 cells had. And in essence, the reason those cells stopped growing is because they were growing in response to growth factors that were in the serum from a tissue culture medium. And they had exhausted the supply of the growth factors in the serum, and so they stopped growing. They didn't know that at the time, back in the day. But what they found is that if they took these cells and removed the serum from the cells, waited a while, they would sort of all accumulate in G0, in the resting phase of the cell cycle. They didn't really get into G0. They got into early G1, and they sat there until they got a new supply of serum, and then they would start to grow again. And that was called cell cycle competence, because they would grow, they would start to move through the cell cycle, but they would not undergo DNA replication and mitosis. They would sort of almost do that. Then they needed something else that was also present in the serum, that they were called progression factors. And ultimately, that part of the cell cycle was progression through DNA replication and mitosis. And the cell cycle people in the 3T3 system had separated those two things. Well, when I learned about this and I was thinking about this whole thing, I said, well, yeah, the T cells do that too. The T cells cause cell cycle competence. They become competent to grow, because when you trigger the T cell receptor, the cells make IL-2 and they express IL-2 receptors. But if you block IL-2, let's say with an antibody to the receptor or to the IL-2 itself, even though they've been triggered by the T cell receptor, they won't undergo DNA replication and they won't undergo mitosis. However, you could circumvent that by just flooding the system with IL-2, and then they would do that. So it was clear to me anyway that the TCR was the competence receptor, whereas the IL-2 receptor was the progression receptor. And because these were two different, physiologically different things that the cell underwent, I figured that whatever was going on in terms of the inside of the cell, they had to be different between the TCR and the IL-2 receptor. So we go through that in the intro. And by this time, there were early biochemical signaling events that had been published and were reported in the T cell system. And it was found that when you triggered the T cell receptor, there was hydrolysis of the phosphatidylinositides, and which would result then in a marked increase in intracellular free calcium, which would then together with protein kinase C, calcium-dependent protein kinase, would phosphorylate intracellular proteins, and that was part of the signaling apparatus. The IL-2 receptor, we knew by then, and I think we went over these papers. In 1986, Julie Stern published that c-Myb, as a proto-oncogene, was activated by the IL-2 receptor, but not by the T cell receptor. There were other of those proto-oncogenes that had been looked at in the T cell receptor system as well as the IL-2 system. And for example, c-Fos and c-Myc were both activated by the T cell receptor as well as the IL-2 receptor. So they were expressed very rapidly, and then they disappeared almost as rapidly. That's what we knew at that particular point in time. So I wanted to focus down on genes that were activated by the IL-2 receptor, because we knew that Julie's paper had shown us that it happens. Now I wanted to discover new genes. I just didn't want to, sort of borrow stuff from the oncogene people, because they were already working fast and furiously on characterizing these genes as to what they did and so forth and so on. And I figured that if I was going to be up there in New Hampshire in the Northeast Kingdom in the Upper Valley, that I should focus on trying to discover new things. So the problem was we needed a method in order to focus down and isolate IL-2 induced genes in the T cell system. At the time, in the 3T3 system, people had asked the same sort of question, and particularly they had focused on the competence genes that they wanted to. They found that the platelet-derived growth factor was probably the competence factor, and the receptor for that was the competence receptor. And so they did differential cDNA hybridization in cDNA libraries to discover new genes. And that worked, so that you would take cells and stimulate them with PDGF or serum or whatever, and then extract messenger RNA, and then make cDNA probes from that messenger RNA, and compare that with unstimulated cells, just resting 3T3 cells. The only problem with that was that you only really could detect very high abundantly expressed genes, greater than 500 copies per cell. And I felt that what was the money was probably in the very rare genes, that you didn't need to have a whole truckload of these things. You could just get away with a few. So at the same time, another group had found in the 3T3 system, again, had found that if you blocked protein synthesis while you were doing this incubation with your growth factor, you could superinduce genes. And the reason that was the case had been shown to be due to the fact that many of the genes that were activated in this kinds of situations were only transiently expressed. They were just like CFOS and CMYC. They come up and then they go back down again very rapidly. They're messenger RNA. And the reason that the messenger RNA would go up and down so rapidly, it was good if you only wanted to have your transcription occurring transiently. You didn't want it to be going on forever and ever. And the way that was regulated is that there were sequences in the 3' non-translated region of these messages that targeted them for degradation and destruction. And so when you blocked protein synthesis, you blocked those proteins that were targeting these messenger RNA copies. So you'd superinduce genes with cyclohexamide. So that was good. You could do differential cDNA constructs. You could block protein synthesis and get superinduction so that you could increase your sensitivity of your whole thing. But the kicker in these experiments was furnished to us by Arthur Pardee. Now, Pardee was world famous in molecular biology circles. And this shows to Goya a little bit how science works. When Doreen Cantrell's paper in Science came out in 1984, that really showed the world how IL-2 and IL-2 receptors caused T cell cycle progression. One of the persons that noticed that paper was Arthur Pardee, because we basically had answered many of the questions that he had posed over the course of 20 plus years working on he started working on this on cell cycle progression in the early 70s while he was at Princeton. But this time he was at the Dana-Farber, and he invited me to come down and give a seminar after Doreen's paper came out in the late 80s or whatever. So I gave the seminar, and then we sat down and talked about life and science and that sort of thing. And we hit it off. It turns out he was, I'm not very tall. I'm only 5'4", and so therefore I like little people. I don't like those great big tall people. I mean, it's not that I dislike them, but just if I had my druthers, I'd have little people. So he was about my size. And he'd also done a postdoc in Paris with back in the 50s when he was a young guy. He was 20 years older than I am. His experience in Paris in the 50s looked to be like almost identical to my experience in the 70s. So we had these common, we could establish these common bonds, and that was good. Well, I decided that what we needed to do we needed to really get down and explain to Art what we had done with the IL-2 and T cell system. And so I figured the best way to do that would be to have him come to. We used to have meetings at the Dartmouth College Menarie Conference Center, which was on Squam Lake. I can't remember whether I've mentioned this before, but Squam Lake was about an hour and a half drive straight east from Hanover, in the foothills of the White Mountains. And this lake was a very special lake. And actually, it's the lake where they filmed the movie on Golden Pond with Henry Fonda and Jane Fonda and so forth. And actually, we were there in the summertime when they were making that movie, and we would go out in a speedboat that they had at the conference center and check out the filming site and so forth. It was fun. So we went to the Menarie Center for 3 days, and we went over everything. And Art told us about a new technique that he had worked out that the yeast cell people had worked on to identify and clone induced genes in the yeast. And Art had adapted it and adopted it for the 3T3 cell system that he published, I think, in 1988. Was where we took off. And that's where we started on these experiments. In terms of the materials and methods in this paper, in terms of just chemicals that we had recombinant IL-2 from Takeda. We had I forgot to tell you that the method that Art had worked out for his system was that you would use precursors of RNA, uridine and guanosine, and that had been labeled with sulfur atoms attached to it. And so those precursors would be incorporated into the newly transcribed messenger RNA transcripts. And then you could purify the labeled messages versus the background of housekeeping messages that were there all the time, and we weren't interested in them. You could, by using a column made out of agarose beads that had been complexed with mercury. And mercury and sulfur, if you know your inorganic chemistry, form a covalent bond. Now, you can't get to a better affinity column than that, because you could incubate your message that had been labeled with sulfur with the beads and into a little column like tuberculosis or a tuberculin syringe kind of thing. And then you could wash everything so that the thiol derivatized messages would form covalent bonds to the mercury beads. You could then wash everything else through. And then you could simply reduce that with 2-mercaptoethanol so that it would reduce that chemical bond between the sulfur and the mercury. And then that would just fall off right into 1 or 2 tubes. So it was good. So we had that, and so one of the other, and back to the chemicals, we had a 4-thiol-uridine and 6-thiol-guanosine. We had the phenylmercury sulfurose that we could buy at the store. In terms of our cells, we had our cell system that I've gone over in detail with PBMCs activated through anti-CD3, and then cultured with IL-2 for 10 or 11 days, and then taken out of the IL-2 for a day and a half, 36 hours. And then we could reactivate the expression of the IL-2 receptor because it had gotten low by then, by activating the cells with a 4-ball dibutyrate, which we could wash out then after a 12-hour incubation period. And then we had synchronized G0, G1, early G1 synchronized cells with IL-2 receptors expressed, and then we could add IL-2, and then we could do that with cyclohexamide or without cyclohexamide and so forth. And that was how we generated messenger RNA to construct our libraries, our cDNA libraries. We would activate the cells in the presence of 4-thiouridine or 6-thioguanosine for only 2 hours, with plus minus cyclohexamide. We would then take the cells, lyse the cells, isolate the RNA through guanidine hydrochloride, routine method. And then we could absorb them onto the mercury columns, and then we could wash the mercury columns to take off any non-specifically bound materials, first with a normal saline and then hypertonic saline, 0.5 molar. And then we could elute the messages that were covalently bound to the mercury with 2-ME, as I've said. And then we could then take those transcripts. We could also label at the same time by using tritiated uridine and so forth. And then we could take those transcript, reverse transcribe them, make cDNA, and then clone the cDNA into plasmids and E. coli and so forth, using the routine methods by then of genetic engineering. Let's see, I think it's time to go to the results. So we will screen share at this stage. And this is figure 1. And it shows you, here's what we did was we labeled the RNA with tritiated uridine. And here is the amount of tritiated uridine bound into RNA that was in the flow through. Here is in the wash, and here's the eluate when we added 2-ME and then brought it all off. And so you can see that basically we purified the RNA by greater than 90%. Then we wanted to see what the message looked like. And so we used c-MYC as a cDNA probe and did northern blots on the RNA from the flow through and from the washes and from the eluate. And you can see that there's a massive increase in the specifically labeled c-MYC. So that was very helpful because it told us that we were on the right track. So then what Carol did over the course of more than a year is that she looked for messages that were activated by IL-2 and also activated and super expressed by cyclohexamide. And she came up with basically 8 new genes that had never been described before, that had not been analyzed like this before with IL-2 and cyclohexamide. So we decided to call these cytokine response genes. This is number 1, 2, 3, 4, et cetera, 5, 6, 7, 8. And so this is no addition. So this is resting T cells, the message. Here's cyclohexamide alone. And here you can see, for example, in CR6, you have a message that's induced by cyclohexamide without any IL-2. Here's IL-2, which is less than that. But then when you have them together, you get a superinduction of this particular gene. And then here's another CR7 and CR8. And that was the general pattern. So that was figure 2. And if we go then on to, so then we took those cDNA from the colonies that we had identified and sequenced the cDNAs to see whether or not any of them were already in the databases. And we found 2 out of these 8 genes, 2 of them. This one, number 4, is called SATB1, which is a DNA binding protein, and CR7 which coded for a gene called PIM1. PIM1 is a serine threonine kinase. So that was good because we had 6 new genes that had never been described before. And this was the amount of IL-2 induction. And you can see that there's quite decent induction, 24-fold, 22-fold. This is greater than 50-fold. And so forth. That was CR5. And so that was very, very, this is exactly what I was looking for, were new genes that we could then characterize. And I think there's just one more, really shows the cyclohexamide effect. And these are now these 8 genes, CR1 through CR8. And you can see, and then these are different times after the activation was started. It was time 0 over here. And then half an hour, 1 hour, 2 hours, 4 hours, and 8 hours. These were, all of these are northern blots using the cDNAs from these different clones. And this is IL-2 plus cyclohexamide onto T cells that activated with nothing and then 30 minutes and so forth. And you can see that the effect of cyclohexamide is quite striking in several instances. There's always a lot more there than without cyclohexamide. So that was a good demonstration of our strategy and our tactics in terms of this. Here's CR6, which was markedly enhanced in terms of its expression. Then we took all these genes and we prepared northern messenger RNA and northern blots after the TCR had stimulated them. And there's no data to show you here because only one, which was CR4, which is the SATB1 gene that was also activated by the T cell receptor. So we had 7 genes, 7 new genes that were clearly IL-2 induced genes. And that's exactly what we wanted. So I'll stop screen sharing. Now, in the discussion of this paper, we established several points. First of all, we said this paper establishes a method to identify and clone cytokine response genes. The thiol labeling procedure increased the efficacy of cloning from less than 10% of the total RNA. And so that was really, really good. That was number one. Number two, the competence and progression genes clearly differ in this particular instance, which is one of the points that I wanted to establish. And in terms of G1 progression lasts about 10 to 12 hours. And so you have early G1, you had mid G1, and then you had late G1 just before the G1 S phase transition. And so you could stratify when you wanted to find the gene expression by this method. And at this point in time, we point out in the discussion that there were a whole bunch of new lymphokines, interleukins that had been discovered by this time, that were newly described cytokines that a lot of times nobody knew exactly how they were, what they were for in immunology and in the immune system and the immune response. And for example, in particular, the same genes were found to be activated by the beta chain of the IL-2 receptor. The EPO was the erythropoietin receptor, and the IL-6 GP130, the signaling chain of the IL-6 receptor, which all those three are very important. Subsequently, were very important. And so we could look at IL-2 in T cells. We could extrapolate pretty much to these other tissues, essentially red cells and so forth. And we then went on to speculate or to conjecture that you could classify the various cytokines that we were all discovering by the receptor's cytoplasmic domain. You could do that with regard to their external domain and which cytokine that they would bind to. But we could also do it with the cytoplasmic domains in terms of sequence homology and so forth, as to whether there were similarities or differences. In addition to that, you could then also look at the biochemical signaling pathways from all of these different receptors. And now, as a consequence of our CR genes, you could look at the genes that are activated by the different receptor systems and signaling pathways. So that's it with this paper. It was, from our vantage point, this is where we'd started to work 20 years previously. So this is a culmination of 20 years of hard work in this whole field. Now, subsequently, all kinds of things happened with regard to this whole story. Carol, when she finished her, so this was basically Carol's thesis. When she finished her or dissertation, and when she finished her PhD work, she needed to go for a postdoc, and I suggested that a good place for her to go would be to go to London and work with Doreen Cantrell, which she did. And so she went there, and I think around 1994, and she had several papers that were just as important as this particular paper that came out of that experience. So this paper that we reviewed today was 93, and in 94, Carol and Doreen published a paper that was entitled Activation of JAK Kinases and Stats by Interleukin-2 and Interferon Alpha, but not the T cell receptor. And as you know, that's very, very important. And as you know, I think I already mentioned in one of the past sessions of the Journal Club, Tada Taniguchi, who was an arch rival for me at that time, had published a paper, I think in 91 or 92, in Science, no less, that the IL-2 receptor activated protein kinase C and calcium flux and so forth and so on, the LCK, tyrosine specific kinase. That was before JAKs and stats had been discovered. That just happened in the next year or so, in 93, 94, by two major groups, one from the Imperial Cancer Research Fund, where Doreen was, with Ian Kerr, so George Stark, and then in New York with Jim Darnell, who had discovered a lot of the stat molecules and the stat molecules. And so JAKs and stats were rapidly jumped on by everybody who was interested in cytokine receptors and so forth. So the race was on at that stage of the game. So these papers that came out from Carol and Doreen were right in the thick of things at that particular point in time. And I had decided that I didn't want to get in because I figured it was going to be the weeds. There were already a lot of people working on this whole thing, and that was a good strategic decision on my part. In 1995, the very next year, Carol, again in Doreen's lab, had shown that interleukin-7 activates the same JAK stats that were activated by interleukin-2 in T cells. And then in 96, Doreen and Carol published another paper showing that the JAKs were necessary for signal transduction through these cytokine receptors, but you also needed serine and threonine specific kinases to activate STAT5. And so that was all of that those were very heady times in this business and in this field. And I must say that the immunologic communities interested in the T cell receptor and that sort of thing sort of ignored it. Oh, it was a ho-hum. And the major players that worked on this field, the JAK stat discovery people, and then it brought in all the hematopoietic cytokine people. So the major players were not the immunologic community. And the immunologic community really didn't follow up on this paper that came out in PNAS in 1993 that Carol had spent several years of her life on. I'll stop there. So don't forget to like the video if you did, and click on the bell, get notified. check out the website and read this paper, and then decide for yourself whether what I've just told you is correct or not. And jump on the bandwagon and come back next week. Thanks for listening.